Now we're going to talk about karyotypes. If you watched my first video, you would already know the answers to the first question. The normal numbers of chromosome in a human somatic cell would be 46, and a normal human gamete would be 23. Why is it 46? Why is it 23? You get one sperm from dad, 23 chromosomes, an egg from mother, 23 chromosomes put together. That's you. Okay. Yeah. Anyway, the 23 chromosomes that are produced by a gamete is done through meiosis, and if there is an error during meiosis 1 or 2 during the anaphase of meiosis, whether it's homologous chromosomes not separating or sister chromatids not separating, then you would have an abnormal amount of chromosomes in the gamete, which will cause an abnormal amount of chromosomes in the child. So what's the diploid number? The diploid number is 46 because human somatic cell is a diploid cell, and a haploid number is 23 because a normal human gamete cell is haploid and it has 23 chromosomes. Now, new word, autosome and sex chromosomes. So if we were to look at not this and not that, if we were to look at a karyotype, such as this one, first off, you are asking, what is a karyotype? A karyotype is, you're looking at a person's cell, not you, exactly, but a scientist would look at a person's cell, and they would kind of freeze the chromosomes in place so that we can kind of separate them and organize them, and we would organize the chromosomes by pairs. Pairs of chromosomes are called homologous chromosomes. It's pretty important. So as you can see right here, we have 23 pairs of chromosomes, just like a normal human somatic cell, and the first 22 pairs, right? See, they're all numbered. The first 22 pairs are called autosomes because they're not sex chromosomes. We give it another name called autosomes. And sex chromosomes are the ones that determines your sex. There are also other types of proteins and traits coded by the sex chromosomes, but for your knowledge, it's important to know that sex chromosomes determines whether you're going to be biologically a boy or a girl. So if there are two sex chromosomes, one is X, one is Y, how do you know one is X, one is Y? Even if I don't label it, the X chromosome is larger than the Y chromosome, and it's the way it is, and that's fine, right? It doesn't mean that if you don't have two X chromosomes, you're going to be there's something wrong with you. There's nothing wrong with you, or you're just a boy. X and Y chromosome together makes you a boy. And then, if there is a person with two X chromosomes, such as this one, right? This is a human female, normal human female karyotype. They're the first 22 pairs of chromosomes, and then we have two X chromosomes. That makes a person a female, okay? So that's a karyotype. So now you know what an autosome and a sex chromosome is. How many autosomes are in a normal human body cell or normal human somatic cell? There are 22 pairs or 46. How many sex chromosomes are there for a normal human somatic cell? There are 2 of them. It's either X and X or X and X or X and Y. Moving on. The next one is how many sex chromosomes. Okay, now we can also talk about a gamete, right? So if you were to look at a karyotype of a gamete, and even if I don't show it to you, you should know how it looks like because you watched the last video. Or did you? So here's a gamete. Ooh, patient 3 died. Anyway, this is a gamete. You know it's a gamete because there aren't pairs of homologous chromosomes, and if you were to count the numbers of chromosomes, there are only 23. 23 must be a gamete, okay? So in this gamete, you can see that there are only 22 autosomes because during meiosis, the homologous chromosomes got split apart, so now you only get one copy of each type of chromosome. So these first 22 are called autosomes, and then you have one sex chromosome right here. Together, you get 23. So the answer for this one is 22, and this one is 1. I hope that's what I said. But this is the right answer. Now, how we're looking at a karyotype because we can determine whether a person is a boy or a girl pretty easily, but for most people. However, if a scientist is suspecting, or a doctor is suspecting that a person might have a chromosomal disease where this person has more or less chromosomes than what is, what is supposed to be without the mutation. It's not saying that there's something wrong with this person, right? It's still a person. It's just, by chance, random chance, this person got an extra few or extra, a few less chromosomes. It really has nothing to do with that person itself. Takeaway for you, right? When you look at a person, I know you don't see people with chromosomal disease if it's very often, but if you were to run into them one day, think to your head what you just learned, right? There are random things that could just happen that would, make you different from other people, and you're different from other people anyway. Even as you, a healthy boy learning at Georgetown Prep, you're still different from other people, and you're just different in different ways, and that's fine. Okay. It might not be fine for some people. They might not be as healthy, or there might be things that they cannot do. But that's not their fault, right? They didn't deserve it. There was nothing they did in their life to deserve that, but it just things that happened. And at the same time, you should feel lucky that there are certain things that could happen that didn't happen to you, that might not be so, so good. You might not want it, but it could still happen, even if you don't want it. Anyway, that's enough, my moral story for you. I think the takeaway, the real takeaway is that I just told you. Anyway, so now we can go back to biology. Now that you know that non-disjunction happens, and sometimes people are born sick, a little more sick than other kids, and it happens. So now, but what is non-disjunction? Non-disjunction happens during meiosis 1 or meiosis 2. So this right here is a really, really good picture. Take a look. This is the before cell. This is how your cell looks like before meiosis. This is meiosis 1, how it's supposed to happen, where homologous chromosomes separate from each other. Meiosis 2, you have sister chromatids supposed to separate from each other, and you get 4 gametes that are all 1N or haploid, but the original cell is 2N and diploid because there are 4 different chromosomes. 2 4X shapes, that's 4 different chromosomes, in case you didn't know. In case you needed a refresher, because you already know. And then non-disjunction can happen during meiosis 1, where the homologous chromosomes don't separate from each other, but they separate just fine. During meiosis 2, then the result is 2 of the daughter cells would only have 1 chromosome, or it will be 1 short, or it could have 1 extra one. So these are going to be called trisomy, and these are called monosomy, I think. Now, non-disjunction can also happen during meiosis 2. So if that happens, you would compare this right here to the original one, right? Let me make it a little smaller. To the original one. And what happens is meiosis 1, homologous chromosomes split just fine, but meiosis 2, the sister chromatids did not separate right here. Oh no. So instead of only having the regular haploid numbers of chromosome, you would have N plus 1 or N minus 1. And that's how you get an extra chromosome. This extra chromosome could happen anywhere within the genome. It happens. All right, you should know how to draw a normal human male somatic cell, but you should also be able to, tell what a certain disease is if I were to give you the information. So let's say Down syndrome, because everybody knows about Down syndrome, you're using it as an example. Down syndrome is when a person has an extra chromosome number 21. This is not Down syndrome. Extra chromosome number 21, okay? So it will look something like, this is a better one. Extra chromosome 21, okay? Then I can give you information, a description of all different kinds of syndromes. But then if one of the information tells you that non-disjunction result in an extra chromosome 21, you would match it with this karyotype right here. I can also give you a karyotype and ask you whether it's a normal human body cell or not. So for example, if you look at this one, this is not a normal human body cell because there's an extra 21. However, if you were to look at this one right here, this also doesn't look very normal because we're missing one right here, okay? So, if you know what a regular a normal non-mutated human somatic or gamete supposed to look like, this is how it normally is supposed to look like, then you would know when there's an extra one somewhere, or where there's one less, then there's a mutation. Okay, that's the end of the video. I hope you will do very well on the test. And oh, that was Marshy's freaking out. You'll see him tomorrow. Okay, bye.